Good morning, class 9. Today we'll take up Kingdom Animalia. Kingdom Animalia includes all the animals. These are multicellular, heterotrophic and motile in most cases. And their cells do not have a cell wall. Children, this kingdom is the largest group including animals which range from small microscopic animals to very large animals like elephant and whales. So naturally, there is a great diversity and variation in different organisms, and the animals can be simple organisms without any tissue or organ differentiation, and on the other hand, there are animals who have very complex level of organization. Therefore, classifying animals into different groups is not an easy task. Different scientists and zoologists have tried classifying them, and the most commonly used system of classification, which is based on the extent and type of body design and differentiation, is that kingdom Animalia is divided into 10 phyla. Singular of this phyla is phylum. So each phylum includes all the animals which are close to each other in many ways. That means they have many common characteristics. So these 10 phyla, they are named as number 1 Porifera, Coelenterata, Platyhelminthes, Nematoda, Annelida, Arthropoda, Mollusca, Echinodermata, Protochordata, and the last one is Vertebrata or Chordata. Before we study about the animals which belong to these phyla, we have to understand some terms used to describe their characteristics. Unless and until these concepts or these terms are not clear to you, you will not be able to understand on what basis are these animals classified. So otherwise it is going to be a very tedious, long and confusing work. So before we start with this classification, today in this session, we will simply just understand the criteria or the basis of classification of kingdom Animalia. Now the different this figure here shows the different bases of classification of animal kingdom. The first one is level of organization, then symmetry, then diploblastic and triploblastic organization or coelom development, segmentation of the body, and presence or absence of notochord. So first of all, if we talk about level of organization, there are although all the animals are multicellular, but they do not have the same pattern of organization of cells. For example, poriferans, they have only cellular level of organization. They do not have tissues or organs. But in some animals, these cells are arranged into tissues which perform a particular function. So they have tissue level of organization. A still higher level of organization is the organ level. So here and in still higher animals like annelids, arthropods, mollusks and all the chordates, they exhibit organ system level of organization. Then second is about their symmetry. Animals can also be categorized on the basis of their symmetry. Some animals are asymmetrical. That means any plane that passes through the center does not divide them into equal halves. For example, sponges. When any plane passing through the center of the body divides the organism into two halves, then it is called radial symmetry, as it is seen in coelentrates and in echinodermates. But in animals like annelids, arthropods, etc., the body can be divided into two identical left and right halves in only one plane. So in this case, it is called bilateral symmetry. As you can see in this figure, this is showing a radial symmetry, the first figure, and the second figure shows bilateral symmetry. Now the third this basis or criteria is the germinal layers, or they are also called the dermal layers. We cannot see these layers separately in an animal, but during embryonic stage, when the animal is undergoing differentiation, these layers are present. And some animals, they have just two layers, that is outer layer called the ectoderm and the inner layer is called endoderm. These animals are therefore called diploblastic. But in higher phyla, the animals have an additional third layer or the middle layer, which is called the mesoderm. So such animals which have three layers, they are called triploblastic. So this was about the layers. Now next is the coelomic cavity. What is the coelom? Coelom is the name given to the empty space between the body wall and the gut wall. This cavity is lined by mesoderm. Animals which do not have coelom are called acoelomates, and the animals which possess this cavity are called coelomates. And in some animals, the body cavity is not true coelom. What is this true coelom? That means the mesoderm, the cavity is not lined by mesoderm, but rather it is present as scattered pouches within the ectoderm and the endoderm. So such a false cavity is called pseudocoelom. As you can see in this figure, the first one is showing a coelomate, second is a pseudocoelomate, and the third one is acoelomate. Then next criteria is segmentation. In some animals, the body is externally and internally divided into segments. For example, in earthworms. So such animals are called the segmented animals, and this is also called metamerism. And the other animals which do not have this kind of segmentation on their body are non-segmented. Then the last is the notochord. The presence or absence of notochord. What is this notochord? It is a rod-like structure which develops in the embryo on the dorsal side of the body. Animals with notochord are called chordates, while the animals which do not have this structure are called the non-chordates. But some animals, they have notochord only in the embryonic stage and not in the adult stage. These animals are called protochordates, and in still other animals, this notochord is replaced by vertebral column or the backbone in the adults. So these animals are called vertebrata, and this vertebrates is the largest phylum of this kingdom Animalia, which is further divided into classes which we will study later in this chapter. So these were the 6 criteria on which this whole classification is based, and now this flow chart which is shown here it is giving a summary of the entire thing whatever we have studied today. These are the different bases. Level of organization can be cellular level, tissue level, or organ system level. Symmetry, the animal can be asymmetrical, it can have radial symmetry, or it can be bilaterally symmetrical. Then if the animal has 2 germinal layers, it is called diploblastic. If it has 3 germinal layers, it is called triploblastic. If the animal does not have the coelom cavity, it is acoelomate. If it has a cavity, coelom, it is called coelomate. And sometimes if they have false cavity, then it is called they are called pseudocoelomates. Then segmentation, animals with segmented body and animals with unsegmented body. Then the presence or absence of notochord. If notochord is not there, they are non-chordates. If it is present, then they are chordates. And chordates, if the chordates are further divided into 2, if the notochord is present only in the embryonic stage, then it is protochordate. But if it is replaced by the vertebral column in adults, then it comes into vertebrates. So this is how the animals are classified. So this in the next video lesson we will study in detail about each of these 10 phyla one by one.